So thank you very much for the invitation, and as I'm all the time very happy to be part of the Capri and CASP community, and yeah, like so a lot of like method development what we are doing in our group is tightly connected. Originally this Capri, but now this also like Yakabili, like working on task challenges and the joint endeavors. And I was asked like by the assessors and Mark to talk a little bit of historical perspective, basically. So how kind of like what sort of we have been doing over some time and how it is evolving during the new times and everything. And so, and as you know, kind of like one of the key tools which we are developing right now with Chandler is something called CLUSPRO. And originally it was initiated by in Chandler's lab by current PPS Professor Carlos Camacho, where basically what his idea was that if we can do like, I mean, for physical problem, if we can do some really, really good sampling, and then we can store it, but it's basically both allocate for the good energy, but also at the size of the nodal that really helps kind of to figure out things. And actually, like the method was developed exactly as one of the early Capri rounds. And so initially it was using some FFT docking programs, actually several of them, and trying to see and basically like, and then the results were scored with some consistent energy function, clustered and trend based on kind of energy and the cluster size. And this idea kind of like works very well. And we have been able to improve kind of the sampling, so changing kind of from multiple programs to one program. And I will talk about the methodology, what we have been doing. And like this is basically like it was working quite well. And but obviously, like when AlphaFold appeared, I mean, it definitely like it combined advantages of basically both solving the individual protein fold structures, but also being very good at dealing with interfaces. Although, as was mentioned today, I mean, there is this element of basically copying existing things, so you not all the time know basically what that thing, but still there is a very good like generalization ability. So basically we need to change, and we have been working on this, and it took us some time, but it looks like, I mean, things are moving forward and progressing. And I will talk about this in my talk today. So can I move to the next slide? So basically the premise kind of, as there was a big discussion today, right? I mean, like there is two major issues with the problem, right? First of all is sampling, and second one is scoring, right? So with sampling, the issue when basically like what tools what was described today, like for example, like multi-seed sampling of AlphaFold 2 and AlphaFold 3, it looks like it indeed can massively sample, but unfortunately it is stochastic. So you really need to sample a lot, kind of and you don't have a guarantee that actually you can cover the space. So what we have been doing is actually doing some sampling method, which basically can systematically cover the space, but with some caveats. For example, I mean, like if protein is not changing conformation too much, we can guarantee that with some level of accuracy we can fully sample configurational space. Of course, the problem becomes difficult if the protein starts changing the conformation. Then you need to look at the ensemble sampling, and it obviously increases the amount of samples, but it's still pretty guaranteed in the sense that you can cover the space, right? And it looks like some of our results indicate that does helps improving of the prediction. And obviously, and I will talk later about it, we're also trying to do it more effectively, right? So can we actually kind of use kind of the systematicity of the sampling space, configurational space, but also do it in a faster manner, like for example, like what modern diffusion models are doing by basically kind of learning like where they should move things more effectively. And that's something what we think about, and I will also try to mention this. So our idea when AlphaFold appeared, we immediately kind of like started to work on this, and there was a very good idea by Dmytro, who is here in the audience, who thought that okay, I mean, like, and then basically many other groups noticed it, that AlphaFold was able to to score the structures, right? That, for example, if you give kind of like some input configurations, you can actually tell kind of whether it is good or not, right? And so we tried this idea kind of with together with docking, where we actually give some of the samples in docking and try to score it. And it did seem like that in some test cases it worked well, but actually in previous CASP, it failed spectacularly. So we tried basically to bet on this idea, and basically we got not much, right? So so that was a lesson for us, and we persisted. We actually thought about kind of what could be the issues and what can get wrong. And one realization what we saw, that basically because the way how AlphaFold is trained, it's trained actually on template of crystal structures and things like that. So the type of structures, and obviously we wanted to do it cheap, right? Like typical CLUSPRO is giving you like 20, 30 models, and some of these models actually are typically what CASP calls or Capri calls acceptable quality. But the problem is that, they're not good enough, they don't look like real crystal structures or cryo-EM structures, basically. So that's why it wasn't super useful to AlphaFold, and so results were actually not kind of, on interesting cases, actually, were not that much improving. But that was a good lesson to us. And we thought, okay, can we actually, does our sampling achieves kind of like these good quality structures? And we sort of embarked on pretty long kind of exploration, basically. So what level of structures could be good enough kind of that it would be like recognized and scored well? And this is basically was work again, it's a number of years work by Ryota, who is here in the lab, who actually figured out that basically we do, we can actually work with something like basically what Capri calls medium quality and above structures. So actually, if we can generate a medium quality of above samples, then actually we can modify and change AlphaFold to actually to recognize it and use and basically help kind of improve kind of the capacity of the method. Okay, so that basically was our kind of ideas, kind of which we had again. We have an anecdotal evidence for this, but it seems like, we were, and actually some benchmarking data too, so we felt it's pretty confident about this, that this is actually something interesting. So another line of work what we have been doing is basically since we've recognized kind of like so basically what we want to make and modify AlphaFold to be kind of like more from global sampling engine to kind of more local sampling and refinement. Actually, a number of years what we have been working actually is some form of refinement using kind of like physics and force field, but that basically wasn't kind of, it was working up to degree, but not super conclusive, right? So here basically we tried and see can we actually sort of do something about, do something about this and the refinement. So we started actually to dig deep inside AlphaFold. So we generated our own kind of toy version, kind of which is basically similar to OpenFold, which can work basically with standard AlphaFold based architectures, but also with capability of training, modifying, playing with confidence, kind of other type of things. And yeah, this work is pioneered by Eric in our lab, right? So who basically who is also here, who actually did a lot of work on this. And what we showed that we can do on some particular tasks and challenges, we can actually do quite well. For example, modeling say MHC type interactions, where we could actually do much better than even than both template based and standard AlphaFold based AlphaFold methods. actually to predict like accurate structure of protein MHC interaction, and actually this was shown in Capri. Like also is good, and basically what we found that actually when we tune the model in particular tasks, actually the model are not forgetting, so there is no catastrophic forgetting, so we can still do, for example, so MHC peptide model, we can quite confidently do MHC TCR model, which we got like actually high quality prediction in one of the Capri rounds, basically like using the tuned model. So yeah, we also obviously, since we're interested in antibodies, we're also tuning kind of like, and since we're interested in physics-based docking and sampling, we need actually accurate conformations of our monomers or pieces what we use for docking. So in case for antibodies, we're actually doing some tuning basically of the model to better deal with that, with the loops, right? And what we see kind of, and what experience shows, it does seems like that advantage of the tuned models, it actually it arrives you to the result faster, so you can in principle get kind of to it with the standard model, but it takes you actually much longer kind of sampling effort to achieve this. And this is actually very useful for kind of speeding up the protocols and making it useful on time. Yeah, we also doubled a little bit with global sampling. So we did kind of some phosphorylate basically models. So we work a lot with some mass spectrometry groups and everything. So we did like a phosphorylation model actually earlier than it was integrated to AlphaFold 3. And we also see it's actually, it's quite good for the tasks that we are doing. So we're also doing some peptide sampling models. So, and like also, I mean, one of the things is, for example, I mean, that since there is some component of memorization, we also decided to play actually, and we trained like a kind of like recent protein interaction data kind of model, because again, the idea is that we want to be able to sample. So basically, like, so we don't need to wait until like DeepMind like updates the parameters, right? I mean, like we can actually deal with this ourselves. So basically, what was our pipeline? So we tried kind of basically again, so we followed kind of again this belief kind of that's basically what we should be doing, but also we followed the lessons of the previous CASP. So clearly, like, I mean, you need to sample as much as possible, and kind of like, so we use basically, I guess, two ways of sample you can recognize as almost, I think everybody kind of who was talking here of mentioning kind of this in the pipeline. So we use basically samples from AlphaFold 3 and from multi-seed of like various versions of also AlphaFolds. Also, as was pointed out right now, like, I mean, yeah, like this, I mean, for example, AlphaFold is not using templates. For example, I mean, it uses only monomer templates. So basically, so you can enable contacts and actually like sort of make AlphaFold to actually treat kind of the full complex as a template, but still manageably refine. So, but basics, and the addition kind of what we have been adding is what we learned, right? That actually you can provide additional orthogonal way of physics-based sampling, because it's still a lot, right? I mean, if you think about it, I mean, you have like billions of conformations of possible protein and orientation even for fixed things, right? So we can drop down it only like to actually after like this medium quality docu clustering to around like maybe 1,000 samples. Yeah, okay. Yeah, so I'm moving. So we can drop it down to around 1,000 samples actually. So it's not much, and you can actually deal with ensembles, right? And basically we decided kind of we are doing kind of, yeah, like here, like hands off, right? So like that basically we just basically get all the samples and we go through like kind of just one kind of like, I mean, basically like the refinement. And again, kind of, as you can see, I started actually with the history, right? And the idea of like original CLUSPRO. And actually it's kind of, I guess, here is somewhat similar, right? I mean, like we're actually getting samples from multiple things. So we are rescoring it basically. So the idea is that what we are getting are conformations which are, either machine learning quite meaningful or physically meaningful, but also potentially of having capacity of being of good accuracy, and checking with AlphaFold confidence basically whether it recognizes that something kind of like what might be good, right? And then and so basically and then we're doing just basically like clustering, also much finer grain, something like Bjorn was doing. So like with basically like with very fine quality. But actually what we found, I mean, the confidence scores sometimes are jumping. So it's better to introduce not a lot, but a little bit of population score. Basically, we do want that actually there are several kind of things converge to one model if it exists. Obviously, if it doesn't exist, it will take the best score. But if there is multiple models, that would prefer it more. And we do see actually it does improve results a little bit. And this is in principle kind of, it's fully kind of automatic procedure. And the only thing what we did for human, apart from one thing which I will describe, we but just sampled more. We basically, whatever remaining time, Ryota like was very persistent. He just basically tried to use up more conformers and kind of churning through it. And just to illustrate kind of some of the difficult cases. Okay, yes, I'm wrapping up, right? Okay, so basically to illustrate, so here is kind of the like one of the difficult targets. I mean, where basically some very complicated ACPF to be complex, I think, and then doesn't fit in an AlphaFold 3 memory. So basically what we did with so the method just took a basically truncated input of the receptor and generated enough samples and produced a good quality results. So what we did with antibodies, again, it was the sampling, we actually did develop special antibody mode for the sampling. So it was sampling kind of like a lot, as you can see here. And again, and the scoring was able to, we were able to refine it and to some really good models. So one thing is important, what we notice, we can recognize only medial to high quality structures. So this acceptable quality it's not really like that helpful. Yeah, okay, so just last kind of last two slides. Can I get one slide? Okay. Yeah. Uh, can I switch how to move it? So let me um we did show actually some can I how okay. Yeah, we did some MD refinement to show that actually we can work with potentially with MD2. It didn't improve results a lot, but still was interesting. And I guess kind of like what we are working on, right? So this is an automated protocol, but actually like the longer you let it run, like the better it gets, right? So we actually trying to do like things on making it useful for people. Why doesn't the switch doesn't works? So basically we try to make it useful for people. So we're working, as I said, like on kind of like doing say faster FFT docking with machine learning correction, where we see we can reduce amount of samples from 1,000 to at least like 2 times less, right? To do things better. And so I guess I can tell more things if you'll be interested, but basically I guess I want to thank the team, and specifically Ryota, who basically was basically dealing so the FFT sampling on the targets, right, and developed kind of the ideas and the method, and also Dmitry kind of actually was overseeing this work and actually came up with the original idea, and Eric who helped with the tuning and generally and making all these models kind of to be more effective, and also kind of the other team members which helped with the development of the tools, and also a special thanks to our collaborators in Ukraine, which have been part of the team, and basically they did work on the machine learning part of the project. And so thank you very much for your attention.
All right, so that's it for the moment. Patrick will come back soon in from discussion, and we will on to hyperspeed just dodgy. Yeah, so I wanted to do a big introduction, but I see that Remedy is ready, and why that's so ready. Take from here. Previous speaker, he's played back later. On it. Yeah, that's.
They are conscious of the fact that it teach me much to, I'm not overrun at least.